Hello, Niranj. It's a pleasure for me to welcome you here to the CELLIMA talks, and thank you again for agreeing to this interview. And I'm looking forward to your exciting conversation with you. And I think it's best if you start with a brief introduction of yourself and your scientific background.
Yeah, sure. Thanks, Stefan, for inviting me here today. I am Niranj Chandrasekaran. I'm a postdoc at the imaging platform at the Broad Institute of MIT and Harvard. I primarily have been working on the JUMP cell painting project. It is a data generation project where we created the largest publicly available cell painting dataset. It's a data set that comprises of both images and extracted features from cells. These cells were treated with over 150,000 chemical and genetic perturbations. So this is one of a kind. There is no such data set available publicly, at least right now. And we are excited to make fun biological discoveries from this data set. So my background before I joined the lab was primarily in computational biophysics, where I studied protein dynamics. But since then, I've made a shift to image-based profiling, and it's been a wonderful shift. I've learned a lot, and I'm very excited to be working on the JUMP project.
Wonderful, exciting, and very interesting. And I think we will listen to some of your new insights from the JUMP CP project as well, maybe during our talk. But let's start with the interview, and let's dive into it. And which brings me to my first question. Niranj, we're talking about cell painting, and what's your opinion? How can cell painting contribute to the identification of potential drugs in early pharmaceutical development? And in your opinion, what are the specific advantages of this technology compared to conventional screening methods?
Yeah, so cell painting and image-based profiling, the approach that uses cell painting in order to make discoveries, these are kind of novel techniques and methodologies that have just started finding use cases uh, in pharmaceutical research, primarily as a primary screen approach. The reason that it works well is because of, I guess, a few reasons. The first one being that it's really high throughput in nature, which would be one of the main things that you might need for a primary screen, because you would want to investigate a lot of compounds or lots of genetic perturbation simultaneously, and then zero in on specific lead candidates that you want to further investigate. So cell painting and image-based profiling being high throughput naturally lends itself to that. And on top of that, the assay is extremely cheap and it's very easy to perform. That makes it wonderful tool in your pharmaceutical discoveries or pharmaceutical research. It's also quite sensitive. So even very small differences between effects of compounds or genetic perturbation on the cells can be identified using this method. And lastly, it's also pretty quick to perform, starting from performing the experiment with the assay, the image capture and analysis, data analysis, and even discoveries. All this can be done sometimes even within a week, depending on the experiment that you're performing. So all these things together make it an exciting technology in pharmaceutical development, and primarily as a primary screen approach.
Mm-hmm. Yeah, I agree with you completely. And what do you think? In which areas of pharmaceutical research has cell painting already delivered promising results? And which areas of application do you think could be developed in the future?
Yeah, I think one of the most common applications of cell painting and image-based profiling has been in MOA discoveries. This approach seems to work really well when we are in the compound space, especially when we are trying to determine the mechanism of action of unknown compounds, or new mechanism of actions of compounds which are already known. But apart from this, there have also been several other applications recently, not just recently, but in the last few years, that have been published, which include things like determining functions of genes, It could be normal functions of genes, or uh, uh, identifying disease states and how you may be able to revert them using compounds or any other treatments. It has been useful in predicting outcomes of some other complex assays, which I think which is one of the most exciting things or applications of cell painting assays, I believe, in the future, where we are trying to predict the outcomes of other complex assays, which may be time consuming to perform, may be costly. There may be so many reasons why it's difficult to perform these complex assays, while cell painting could be a really good stand-in. It could be a really good substitute for these assays. And you may be able to perform these very quickly, as I said, within a week, and gain knowledge about the system really fast. So these are the applications which have some proof, but also these are the ones that we should look forward to in the future. And in the case of the JUMP dataset that I talked about previously, these are the kinds of applications that we are interested in exploring and making new discoveries.
And nevertheless, are there challenges and limitations associated with cell painting techniques? How can one overcome and improve effectiveness and reliability of results if there are?
Yeah, so I think one of the main drawbacks or the main limitations of this kind of approach is that not every chemical treatment or genetic perturbation in the cell is going to have a morphologic change in the cells. So this method may not be general enough to be applied to every single compound that one might want to study. So that could be a big limitation on this step. There is no easy way to overcome this, so definitely, but this is something that one should keep in mind when using image-based profiling as a part of their experimental workflow. But the other challenge could be that the sensitivity of the experiment, which I mentioned previously, it could be good, but at the same time, it can also pose some technical problems. For example, very small differences in experimental conditions or batch effects that can really be visible in cell painting and can overpower the signal in the data. So standardization of protocols, standardization of equipments across different places or experiments can often be critical in order to make sure that the signal in the data is amplified. And also developing computational methods in order to deal with these experimental artifacts is also crucial. And there's a lot of work that is happening in this space right now. And I'm hoping that in the next couple of years, we will have these standard methods that can eliminate these technical artifacts, and therefore we'll have a higher signal in the data and we can make discoveries with them.
You already mentioned that you've set up a large data set with cell painting, and this was due to the progress that has been made with regard to automation and scalability. How could this be achieved so that screening tasks in early pharmaceutical are now feasible to do with this approach?
Yeah, in recent times, there has been lots of advancements both in the wet lab aspects of cell painting assay and experiment, and also in the data analysis components of it. Of course, we now have robotics that have really automated the wet lab aspects of the experiment. It's not unique just to cell painting, but definitely for all kinds of wet lab experiments, we do have that, and cell painting can benefit from that as well. We also have automated microscopes these days, which can easily capture images from these experiments as well. And then the advancements in deep learning has really made analysis of these images very quick and easy. Deep learning methods have specifically been powerful with segmentation of cells and also with feature extraction. So definitely all the advancements in the last few years have sped up image-based profiling further on top of already it being a fast method. Now, these advancements in robotics and deep learning have accelerated or sped up the performance of, sped up the time it takes to perform these experiments, and therefore its use in pharmaceutical research.
Mm-hmm. And so It's been used in early pharmaceutical research, drug discovery, but what are the changes and challenges that now arise for collaborations between pharmacists, toxicologists, pathologists, and oncologists through the use of these cell painting technologies? And how can these areas be successfully integrated to achieve the greatest possible benefit when using or when going with cell painting approaches?
Uh, like communication across the fields is probably the biggest challenge. That was, I guess, the biggest challenge that we had when we were working on the jump data set as well and production of that, because it requires expertise from so many different fields and people from different fields to come together in order to be able to generate the data set, design the experiments, generate the data set, do the analysis, and also do data interpretation. But given that so many different steps are involved and so much expertise from different fields are required, I feel that all these people working together is definitely something that helps with uh, getting the best out of a cell painting data set. Like we require expertise from cell biologists, we need geneticists if you're working with genetic motivation experiments. Of course, we need chemists if you're studying chemical compounds, microscopists in order to capture the images. We need image analysts to process the images and data scientists to analyze the data. So definitely, we need people from several fields to come together in order to be able to analyze this, and collaboration between them is definitely necessary and also makes it important for being successful and achieving the most greatest possible benefit.
Mm. So cell painting relies on machine learning or and artificial intelligence, meaning that these technologies are interacting and synergizing each others. Um, and how does these technologies have improved efficiency and accuracy when identifying potential compounds and drugs, in your opinion? Or are there already data you can share?
Right. So definitely machine learning and artificial intelligence are really fast evolving fields. And in recent times, whenever there has been advancements in computer vision and computer science, people have been able to immediately incorporate and integrate that with biological experiments. And cell painting is a field, or cell painting and image-based profiling is a field which is sort of at the intersection of computer science and biology in that way. So advancements in machine learning and artificial intelligence immediately end up in image-based profiling experiments for both data analysis and also for all other aspects of the experiments as well. And in recent times, as and when new methods develop, we find that they quickly outperform the old methods that were in existence. Um, Especially in the field of image segmentation and feature extraction, the advancements in deep learning has been significant, and these advancements have begun to outperform the classical approaches of segmentation and feature extraction. Increasingly, papers are now being written where people use only the deep learning based approaches and not even use the classical segmentation feature extraction approaches. And so the advancements have definitely helped in those steps, but also advancements have helped with some of the downstream analysis also. For example, discovering links between how which compounds affect which genes, or links between genes and compounds is quite a challenging problem, especially in cell painting data sets. We have been trying to work on it for a while using classical approaches without a lot of success. But we find that use of deep learning in order to find these hidden links has been much more successful. And we're hoping that the next year or so, there'll be a bunch of papers which will be discussing this in further details. So definitely the advancements are translating immediately into this space and helping with biological discoveries.
You mentioned genetics, and this brings me to the next question: what role plays the integration of cell painting data with other data sources, such as genomics, proteomics, even metabolomics, so that you can obtain a comprehensive understanding of the targets you're interested in and also the mechanisms of action that are involved?
Yes, so there have been a few data sets in recent times which have involved which have both aspects, the cell painting aspects to them, and also some other omic data set also being a part of it. And we find that there is a lot of complementary information between the cell painting or image-based profiling data sets and other omic data sets, specifically genomic data sets and proteomic data sets. And what we find is that the integration of both these like different kinds of omic data set together seems to, in general, increase the power of the profiling approach itself. Um, there's there are some aspects which are well captured using images, while there are other aspects that genomics outperform cell imaging-based assays. But the integration of the two seems to be outperforming those methods individually. So definitely having simultaneous data from different omics methods for your experiments is definitely a good thing to have.
Wonderful, very interesting and exciting. How can cell painting contribute to the development of personalized medical approaches and better predict the efficacy of drugs for individual patients?
Yeah, that's a good question. I feel that the high throughput nature of cell painting and image-based profiling, plus the cost, which I mentioned, and the sensitivity of the method, I think will find a natural role in better predicting efficacy of drugs for individual patients and in personalized medicine. I don't have a lot of particular examples to share at this moment, but I'm fairly optimistic that given the strengths of cell painting, it will end up serving a greater role in development of personalized medicine.
Yeah, you already mentioned you need large data, and now we have personalized data. What role do these large data volumes in data security play in the use of machine learning and artificial intelligence in pharmaceutical research, in academic research?
Yeah, so the large data volumes, of course, are indispensable for machine learning and artificial intelligence applications, both in pharma and in academic space. At this point, I guess I need to stress the importance of having public data sets, because there are large data sets, but many of these are proprietary. But having public data sets will definitely enable a wide range of people, both in the academic space and in pharma, to be able to use these kinds of data and to make novel discoveries. So here's a plug, I guess, for the JUMP data set, and I invite people to make use of this data set to train their models and make biological discoveries.
Mhm. This brings me already to my last question, which somehow is it's not only one question; it's independent three questions. So taking all this information together, all the efforts you have to go for cell painting, to use cell painting efficiently, is the use of cell painting combined with machine learning and artificial intelligence in pharmaceutical research justified? What are the main achievements that have already been accomplished? Just a recap. And what, in your opinion, are the future aspects and challenges that do you see for the successful integration of both technology or all three technologies into the early pharmaceutical research process as a general standard?
Yeah, to answer the question whether cell painting, the integration of cell painting with machine learning and AI is justified, I guess a few years ago, I would have said that it's not there yet because the application of AI in cell painting was still in its early stages. But in recent years, based on all the results that we have been able to see, it definitely looks like that is the way forward. It's still not there yet. We are able to outperform the classical methods, but not by a significant margin. So definitely, it's going to take some more time to have better models and better ways of training the models, and also better use of the models in order to be able to convincingly say that machine learning and artificial intelligence being integrated to cell painting data set is a good thing. So I'm fairly optimistic that it's going to happen in a few years' time, but we are still in that transition state. I guess regarding the main achievements that have been accomplished, yeah, some of the things that I highlighted previously, which is mechanism of action prediction, is something that this method has been really successful in being able to predict. But the one application that I'm very excited about, which I also mentioned, was predicting the outcomes of other assays, especially in the case of, let's say, something like a toxicity assay. So you may not be need to run experiments in, let's say, animal models or something, and you may be able to discover a lot of essential biology related to toxicity in cell systems in vitro, and that could really be beneficial in avoiding having to test all these toxic compounds in animals. So there could be a lot of positives that could come out of that. So I'm quite excited about that in the future. Um, I guess the last question was about the future aspects and challenges of this particular approach. I guess the main challenges right now, the way I see it, that we need, in order to be able to use artificial intelligence and deep learning in self-learning data set, we do need these large data sets, high quality data sets. The jump data set is one such data set, but it may not be sufficient. We may need a much bigger one in the future. So it may be nice to have some of these publicly available data sets and people putting an effort towards releasing these things publicly so that we are able to train better models. And at the same time, another thing that I mentioned, which is the sensitivity of the method, it can be a boon, but it also can be a bane because so many technical artifacts and other things can plague the data set if the experiments are not performed in a consistent manner. So developing methods, the field working together in order to develop methods to tease out the signal from all the noise will be really helpful in order to be able to make interesting pharmaceutical research. So I feel like that is a challenge for the field, and I hope that we are able to find ways around it.
Yeah, let's keep fingers crossed, yeah. But I'm also convinced, like, in agreeing with you. Niranj, thank you so much. So we've indeed reached the end of the interview already. Thank you so much for your exciting insights into this very exciting topic of cell painting, artificial intelligence, machine learning, and pharmaceutical and research. I wish you a pleasant, from my perspective, evening, from your perspective, day. And I would also like our viewers for staying with us so long, and wish them also a happy day or a great evening. Take care and goodbye.
Thank you, Stefan.